We're continuing with life science and cell biology in a nutshell. We're learning about the genetic material and what goes on inside the nucleus of cells. So DNA, we learned, is a double helix molecule. So it has two sides that look like the sides of a ladder, and these are sugar phosphate. The rungs which connect those two sugar phosphate sides are base pairs, which are either adenine, cytosine, guanine, or thymine. And the way the molecules are, they twist themselves into what looks like a helix shape. So also we learned off the strands of DNA, you have mRNA, which is transcribed, and the only difference between mRNA and DNA is that one of the bases, thymine, is replaced by uracil, and also that one of the sugar and the sugar component is ribose in RNA instead of deoxyribose, which is in DNA. That connects, that's in the sugar phosphate chain. So, and also RNA is continuously broken down and replaced, enabling the cell to change its pattern of protein synthesis. Apart from mRNA, which encodes proteins, other classes of RNA are also made by the nucleus. These include ribosomal RNA, which forms part of the ribosomes and is exported to the cytoplasm to help translate the information in mRNA into proteins. Ribosomal RNA is synthesized in a specialized region of the nucleus called the nucleolus, which appears as a dense area within the nucleus containing the genes that encode rRNA. This is also the site of assembly of ribosome subunits from rRNA and ribosomal proteins. Ribosomal proteins are synthesized in the cytoplasm and transported to the nucleus for subassembly in the nucleus. So RNA subunits and these proteins combine to make ribosomes. The subunits are returned to the cytoplasm for final assembly. Another class of RNA that we have is called Trna Transfer Rna. This is also synthesized in the nucleus, which serves as an adapter matching individual amino acids to the nucleotide triplets of mRNA during protein synthesis. The synthesis of RNA is performed by enzymes. So enzymes called Rna Polymerases. To polymerize means to make a chain of multiple units or polymers. In higher organisms, there are three main rNA polymerases, 1, 2, and 3, or A, B, and C. Each is a complex protein consisting of many subunits. One synthesizes 3 of the 4 types of rRNA, called 18S, 28S, and 5.8S RNA. It is active in the nucleolus, where the genes encoding these rRNA molecules reside. Rna polymerase II synthesizes mRNA, though its initial products are not mature RNA but larger precursors called heterogeneous nuclear RNA, which are completed later. The products of the third polymerase includes tRNA, and the fourth RNA component of the ribosome called 5S RNA. So those are the polymerases, which are proteins helping with the formation of our RNA molecules. All three polymerases start RNA synthesis at specific sites on DNA, proceed along the molecule linking selected nucleotides sequentially until they come to the end of the gene and terminate the growing chain of RNA. Energy for RNA synthesis comes from high-energy phosphate linkages contained in the nucleotide precursors of RNA. Each unit of the final RNA product is essentially a sugar, a base, and one phosphate, but the building material consists of a sugar, a base, and three phosphates. So that's where the energy is coming from, is the phosphates. During synthesis, two phosphates are cleaved and discarded for each nucleotide that is incorporated into RNA. The energy released from the phosphate bonds is used to link the nucleotides. The crucial feature of RNA synthesis is that the sequence of nucleotides joined into a growing RNA chain is specified by a sequence of nucleotides in the DNA template. And the pattern is that each adenine in DNA specifies uracil in RNA, each cytosine specifies guanine, each guanine specifies cytosine, each thymine specifies adenine. In this way, the information encoded in the genes is transcribed into RNA for translation by the protein synthesizing machinery of ribosome. Which is in the cytoplasm. In addition to specifying the sequence of amino acids to be polymerized into proteins, the nucleotide sequence of DNA contains supplementary information, short sequences of nucleotides determining initial site of RNA polymerase, specifying where the RNA synthesis should begin and end. In the case of 1 and 2 polymerase, the sequences specifying initial sites lie just ahead of the genes, and the equivalent information for RNA polymerase 3 lies within the gene, within the region of DNA, to be copied into RNA. The initiation site is called a promoter. So the site on the DNA, or the segment of DNA where the initiation begins of the transcription. The promoters of different genes have some nucleotide sequences in common, but they differ in others. The differences in sequences are recognized by specific proteins called transcription factors, which are necessary for the expression of particular types of genes. The specificity of transcription factors contributes to differences in the genetic expression of different types of genes. So these proteins are able to know which parts of the DNA need to be transcribed and which don't, etc. During and after synthesis, mRNA precursors undergo complex series of changes. Before the mature molecules are released from the nucleus, first a modified nucleotide is added to the start of the RNA molecule by a reaction called capping. This cap later binds to a ribosome in the cytoplasm. The synthesis of mRNA is not terminated simply by RNA polymerase detachment from DNA, but by chemical cleavage of the RNA strand. We'll continue in the next part.